Everyone. Thank you for joining us this evening. Our webinar tonight will have two topics. First, we'll be learning more about Combined Brain from Dr. Terry Jo Bichelle, CEO of Combined Brain. Many of you may be aware of the information in our most recent newsletter that the Mowat-Wilson Syndrome Foundation became a member of Combined Brain earlier this year. The second topic of our webinar will focus on our matrix registry. As we go through the webinar, please post your questions to the chat and we'll try to get to all of those. And if there are some that we don't get to, we will get answers and email those to you. So to get us started, it's my pleasure to introduce Dr. Terry Jo Bichelle. He worked primarily as a public health nurse midwife until her youngest child, Lou, was diagnosed with Angelman syndrome in 2000. He quickly switched focus to move bench research into the first clinical trials for Angelman syndrome and to help design natural history studies. Dr. Bichelle earned a PhD in neuroscience from Vanderbilt University in 2016, studying gene-environment interactions in Huntington disease rodent models. He was the founding director of the ABOM Alliance in 2016 and served there until 2018. In 2019, Dr. Bichelle launched Combined Brain, a pre-competitive consortium of patients advocacy groups working to identify outcome measures and biomarkers for rare genetic neurodevelopmental disorders. He was a member of the National Academy of Sciences Committee on process to evaluate the safety and efficacy of drugs for rare diseases in conditions in the United States and the European Union. She is the vice chair of the Tennessee Rare Disease Advisory Council and teaches a course in translational neuroscience at Vanderbilt University. As a parent, Terry Jo has been alongside her son Lou many times as he has
I don't know if you
participated
can.
in clinical issues in the research for treatment for Angelman syndrome. Welcome, Terry Jo.
Thank you so much. And right when you were talking, my internet cut out. So I couldn't tell if you kept talking or not. So I hope it doesn't happen again. But we'll just persevere and if something does happen, maybe Jeanette can text me and let me know, and then I'll know that there's nothing happening. So thank you so much for the introduction. I'm really excited to be here today. And we are excited at Combined Brain to have you all become a member. So I'm just going to tell you a teeny bit about me, just to put it into context. So as Al said, I used
Oh, she cut out. Well, Al, we could go to your slides while Terry Jo tries to reconnect.
Sure, that would be fine. All right. So I wanted to talk a little bit this evening about our matrix portal and patient registry. What I have on the screen is the page that you would go to on our website. Jeanette, Terry just back. I don't know if I should keep going. I know we might have some time challenges.
well,
Terry Jo,
I did just switch to my hotspot, so hopefully that'll work.
Okay. Why don't we go back to Terry Jo and
Okay, sorry about that. I will share my screen again. And I'm really sorry. It was working fine a minute ago, but you know how these things go. So, at any rate, I was working in the country of Mexico and I was carting my little baby around everywhere with me as all the midwives in the country were doing. And we knew there was something not quite right. And it was while I was down in Mexico that we finally received our genetic results. And we were told that Lou has Angelman syndrome. So he was only 16 months old then, and that was actually very young to be diagnosed with Angelman in those days because you had to ask for a specific gene. You weren't able to do whole genome sequencing or a panel. And Angelman syndrome is a lot like a lot of other neurodevelopmental disorders and a lot like Mowat-Wilson in many ways. The kids with Angelman have severe intellectual disability, communication disorder, gross and fine motor issues, sleep disorder. The good news, and I read this about Mowat-Wilson also, is they have a very happy demeanor. So thank goodness for that. Right after I got that diagnosis, I found out that the very first conference ever was going to be held in Finland just a couple of weeks later. So I jumped on a plane with the baby and my mother, who has always been a champion for our family. And we went to Finland. And in Finland, I had the honor of meeting Dr. Art Baudet, who really, from the very first time, he's the person who actually discovered the gene for Angelman syndrome, UBE3A. And from the beginning, he thought that Angelman syndrome was a curable disease. That was radical thinking 25 years ago. In fact, people threw us off of committees when we talked about curing Angelman syndrome. He had some ideas back then about how we might be able to do it even with mega doses of supplements like metformin and creatine. That did not work, but we gave it a try. And we tried a lot of different shots on goal. All of these different options that we tried with Angelman syndrome had been shown to work in mice and reverse their symptoms. And so we were pretty excited. There was a methylation trial, a hypermethylation trial, an L-DOPA trial, which is a drug for Parkinson's disease that had been shown to help the mice, and a minocycline trial, which is a skin antibiotic that had been shown to do something. None of them worked. But what we found during that whole process, there were a couple things. One thing we found is we need more natural history. It's a very big deal and very important, and that's what makes me so happy about you all having your registry up. And at that point, I started to think maybe there was something wrong with academic science, that maybe the academic science wasn't focused on the things that I thought they should be focused on. Maybe they were too focused on basic science and not enough on translational. In other words, not enough on science that could help humans. And so I went back to school myself to get a PhD as a middle-aged lady with 5 kids. And I managed to get a PhD in molecular neuroscience. I graduated in 2016. So it was a big adventure getting that PhD. I ended up studying Huntington's disease as well as Angelman circadian rhythms. And I learned a ton. And during that period of time, while I was studying to be a neuroscientist, other neuroscientists found fantastic ways to treat Angelman syndrome. So by the time I actually graduated, what we really needed were not more compounds. We needed biomarkers and outcome measures. So this is what my son Lou looked like by the time I graduated with my PhD. And during that period of time, Dr. Baudet, the same one I had met right after Lou was diagnosed, he had actually, his lab had come up with a way to actually turn on the dormant gene that was on the paternal chromosome. That's a whole nother story. Anybody wants more details on that, let me know. And that was through an antisense oligonucleotide. And so, in fact, right now, this is a picture of my son this year. He is in a disease modifying clinical trial receiving an antisense oligonucleotide intrathecally every 3 months. It's exciting. It's fantastic. I'm really happy that he's in that trial. But it took us 24 years to get there. I don't want it to take 24 years for you all to get there. I didn't want it to take 24 years for any of these people to get there. And so what I wanted to do was form an organization that could help fast track all these other disorders, including Mowat Wilson. And actually, we have 118 disorders as part of Combined Brain now. I wanted to find ways that we could pool our resources, make sure we didn't repeat anyone else's mistakes, and get to the point where you could have your kids in disease-modifying clinical trials as fast as possible. So one of the things we did, I've done a bunch of Angelman science along the way. I've been very lucky to be part of it. But at this point, I really don't do a lot of Angelman work. I more work on all these other disorders. We did publish this paper last year, at the end of last year, and it's actually citizen science. This paper was the very first paper ever to describe what actually people with Angelman syndrome die of. And in fact, this was not done by clinicians. We worked closely with the clinical units in this country and in Australia to try to document what they could find out about what people with Angelman syndrome passed away from. And while we were collecting 220 cases on Facebook through the Citizen Network, those clinicians only collected 6 cases. So in fact, this paper really pulls in the citizen science more than it does the clinician science. And that's really important. That shows you how much you can do, how much power you have and access and information that you have at your fingertips to be able to do science and change the world. This paper came out in 2022, and it's a what you call a well, it's a cloud that shows all of the science that had been done about Angelman syndrome to that point. And way over here in this little bitty spot is my name, Bichelle. So am actually a part of the Angelman science, and you can be a part of the science for Mowat Wilson. So why we formed Combined Brain, our mission, we're a nonprofit. Our members are all other nonprofits, just like your foundation. And the whole idea is that we can help each other get faster and better science done and get it into people with these disorders. So, one of the main things we found out, and a little bit you could tell from the story that I just told, we did a great job curing mice, a great job curing mice. We tried all these different compounds, like I told you before, and the mice were in the center of that whole effort. And we would get the compound into humans and it would fail over and over again. So, what we realized, and not just we, the Angelman community, really the whole field of translational neuroscience, and including the FDA, what we realized is we actually have to put people in the center of the equation. We actually have to put patients first. So we have to find out what matters to patients. Then we have to collect natural history on families. And then from that, we can go look at the mice or other animal models and see if there's anything that matches what we see in humans, not the other way around. That tells us what endpoints to look at in clinical trials. And that means we're measuring the right thing when we finally get the drug to the FDA, and that means we might have a successful trial. So that's one of the most important things that Combined Brain does. We try to put the patients in the center of all of drug development for these disorders. The FDA also thinks this is very important. So the FDA has actually listed a lot of guidances. They've published guidances that encourage patient-entered and natural history data to be used, hopefully, if enough is collected and if it's done well, it could actually be used in controls for clinical trials. So it hasn't been done very much. It has been done, but not as much as it could be. But that isn't because it's not a good idea. It's because a lot of pharmaceutical firms just haven't started doing it yet. And so the more data you collect, the better data you collect for your registry, the more of a possibility that you can use that data for controls as well. So in general, this is what Combined Brain does. We do every single thing on this slide. So what we do is we start with trying to identify the gene and get people diagnosed, um, help each group come up with their right disease model, which could be a cell, could be fish, could be frogs, could be mice, could be rats, could be all different kinds of models that might be the right one for their particular disorder. In the meantime, we help groups collect natural history that's relevant and what matters to you. During this period of time, the scientists start to recognize what the mechanisms are and how they could actually maybe do something about it. Bing! This is when somebody has an idea for how the disease could be treated. At that point, it's really time to identify biomarkers. It's important to do disease concept models and disease burden studies. I'll talk a little bit more about that in a minute. And that's when it's important to come up with patient-centered outcome measures. I'll talk about those too. All of these things sound really very deeply boring, but let me tell you, they are crucial to getting a drug to clinical trial through the FDA. They're also going to need to try the drugs in other animal models, usually for safety, and then design the clinical trial and then get to the FDA. So basically, everything on top of this line is often the responsibility of foundations. And everything below this line is often the responsibility of scientists and pharmaceutical firms. But you can't do any of it without the other. You can't do one without the other. So you're going to need to have the everything on top also communicating with everything on the bottom and vice versa. So these are all the things that we do at Combined Brain. And this slide is on our website if anybody's interested, which this is the website here. And you can learn more about the different activities that we do. We have a Rent a Brain program where some of our PhDs and pre-medical students can help get science done for each of our member organizations. We have diagnostics, we have a biorepository, we help people do natural history, and we come up with outcome measures. And I'm going to talk about these more in just a sec. So we are really in favor of helping people get diagnosed, and universal newborn sequencing is coming, but it's not coming fast enough. When this happens, every baby born will have whole genome sequencing. It's already being done in New York and in a couple of hospitals in California and around the country, but it's going to be a long time till it's commonplace. So we have a program to diagnose infants. We have not yet diagnosed someone with Mowat-Wilson. But if you know of anyone who's not sure if they have Mowat-Wilson and they need whole genome sequencing, these are all the different disorders that we have diagnosed. We actually take insurance and we have a website where we do an evaluation of the patient. Uh-oh, am I frozen again? Oh no.
We can still hear you, Terry Jo.
Oh, good.
It's
Okay,
just your
good.
video is frozen.
Oh, that's fine, as long as I'm not sticking my tongue out or something. So this is our algorithm for diagnosing infants. It actually spells find out. And so these are all of the different symptoms we look for in infants that we will then offer whole genome sequencing to them and an online evaluation. So after you get diagnosed, what do you do next? Well, we have a biorepository. We started it in late 2021. It took off like gangbusters. At this point, we already have actually closer to 30,000 samples in the biorepository. We collect these samples all over the country. So if any one of our groups is having a what we call a road show, where we send our biorepository team to a location, if any of our groups is having a road show, anyone with Mowat Wilson can walk into one of those locations and do a biorepository donation. We do the consent forms. We tell patients everything about what might happen. And then the samples, all of these different kind of samples get collected, all these different biosamples. We collect data on the matrix, so you'll already be collecting it on your matrix. We can even make stem cells, and we also collect EEGs. And if someone unfortunately passes away, we can actually arrange to have postmortem tissue collected. So anyone can, in the community, can donate with the permission of your patient advocacy group, and they'll tell you how to do this when the time comes. We can even send a phlebotomist to your house. We also can make stem cells, and that would be up to your patient advocacy group, to your foundation, to decide which stem cells need to be made. They would decide that based on knowing which variants, gene variants, or which phenotypes are in hot demand by researchers and industry. And so they would know which lines would need to be made to be able to further the research. None of this is free. All of this costs money. And so the foundation has to, carefully decide what to do. Okay, so now you've got your biorepository and you've got your samples in there. Now you've got to come up with biomarkers. How do you study biomarkers? We can study blood, we can study tissue, we can study EEGs, all of those things at the same time that you're contributing your natural history data. We can try to see if we can tell the difference between people who are more severely affected and people who are less severely affected from the blood, body fluids, tissue, or EEG type measures. And if we can do that, then that's something that we might be able to use when you go to a clinical trial. So if you have a drug that might be helping for Mowat Wilson, how do you know if it's working? At the beginning, it might be kind of hard to tell. Maybe the person's neurons are working better. Maybe the networks in the brain are working better, but maybe it might not be immediate that you see something that changes in the person. So you need to be able to see some biomarker, objective marker that tells you that something is working. A lot of these projects that we do biomarkers for are pre-competitive. What does that mean? That means that we have a lot of companies pitching in funds to be able to do these biomarker projects and then sharing all the data between all the different companies. Or professors and academics and researchers, maybe they can do a pre-competitive project. Or maybe 5 different organizations, foundations can group together and do a pre-competitive project. What that means is that everybody works together, pools their resources, it costs less, and everyone shares the information and data. We also have a cerebrospinal fluid pre-competitive project, which is very important. If anyone in your community is ever having some kind of a procedure, some kind of a brain surgery or something in which they're going to have access to cerebrospinal fluid, we don't do it just for this. It has to be because they're already having a procedure. The foundation can call us and arrange for us to send a kit to the surgeon who's doing that, and they can collect a sample and put that in the biorepository. Those are really valuable because you might be able to measure a treatment effect faster in the cerebrospinal fluid than any other biosample. As I mentioned before, we also coordinate with postmortem tissue collections. This is always a sensitive topic and people don't want to really think about it, but I have already signed up for this. God forbid something should happen to my son. I want to make sure that his tissues go to further research. So this is something that Combined Brain has coordinated. We're also collecting EEGs. We have an EEG bank, and we're putting those EEGs on the matrix. So all these things are actually going on the matrix. And we're just starting this, but as we get more and more and more EEGs on the matrix, we'll be able to do biomarker studies on those EEGs. So if you have gotten an EEG at some point in the past, it doesn't matter what kind, when, how, if you can get a copy of it, and we do have research coordinators that can help you figure out how to get a copy of it, which is part of the problem. It's hard to do that. If you can get a copy of it, you can actually upload that to the EEG bank, and that's almost as valuable as blood and cerebrospinal fluid. So we've already talked about diagnostics. We've talked about the biorepository. I've started to talk about natural history, and that's where Matrix comes in. There are a lot of platforms that collect natural history. We've vetted all of them before we settled on the Matrix, and we try to collaborate with all of these platforms because some of our groups use these other platforms, so we do try to collaborate. We try to make sure that we're collecting as many of the same data points as we can so that we can compare and share data. But we really like the matrix. And the reason we really like it is because it met all the criteria that we cared about. So I'm really excited that you all are planning to use the matrix. These are the surveys that we currently have on the Matrix portal. You can see here, there are a lot of surveys. And if you are a parent, you hate surveys. You have to do them all the time. It's no fun. It takes time. It's a pain. And right when you think you've caught up on all the surveys, it's time to do the surveys again. Let me tell you, this is the most valuable thing you can do for Mowat Wilson Science. Filling in these surveys, and you can do it from your phone. You can put a shortcut to your Matrix portal on your phone. And so if you're waiting in line somewhere, you can fill out some of the surveys. If you're watching some boring TV, you can fill out some of the surveys. These surveys that you fill out, this is your power. This is where you are in control of this data. You can also do something really interesting on the matrix, and this might take a little time for you to learn how to do, but you can set up individualized measures for your own kid. So let's say your own brother, sister, or kid has a particular issue that you care about the most. So my kid with Angelman, he falls a lot, and I worry about that, and that isn't collected by the clinicians. I want to keep track of all the times that he falls, and I want to see if the treatments that he's getting are reducing those number of falls. You can set that up individually on the matrix and keep track of that. And so what happens if you look at this right here, what you can see is this person was keeping track of being shaky on their feet, being sick, taking a nap in the daytime, which is weird behavior for this person, sleeping well at night, which is weird behavior, falling, which happens a lot for this person, developing in a new skill. And so what you can see is, in fact, you can guess, because you can graph all this stuff, this person got a new treatment at the end of June. So what you can see is that this person doesn't have any more episodes of shaky on the feet. They're sleeping better and they're developing new skills. It's something that could be really, really important in the future. Okay, I'm almost done. We also do observational trials through Combined Brain. And that is, let's say, that your physician says to you, I really think you should start taking vitamin B12 because I think there's some evidence that that's really helpful for Mowat Wilson. I'm making this up. I have no idea if that's a good vitamin. So don't take vitamin B12 just because I'm saying this. But you want to try that and you want to see if it's really working or not. You can collect data before you take that, while you take it, and after you take it. You want to coordinate that with your foundation. But that is something that you can do. And then if you get enough pilot data, you could study it and you could publish it. Just to speed through the last bits, we've been developing with a team at Duke University this measure called the Observer Reported Communication Ability Measure. It was developed for Angelman syndrome, so we borrow as much as we can from what other people have done. And we are then expanding it to all of these disorders with the hope that eventually it can be used in all neurodevelopmental disorders. We're also we're in the middle of creating ourselves a toileting ability survey. That's because we talked to a lot of families, and they said if you could do one thing with a new drug, you would help my child be able to be independent at toileting. So we wanted to see if we could measure improvements in toileting abilities. So we have this thing called the ORTUS, the Observer Reported Toileting Abilities Survey, and that's something that we'd love everyone's help on. So all of this is based, all the work we do comes out of FDA guidances. We read these FDA guidances like they're instruction manuals for everything we're supposed to do, and we follow those so that you can actually use all of this data that you collect and everything you do in clinical trials in the future. And one of those is to do a disease concept study. A disease concept study is different from the kind of studies that you do with clinicians and even in natural history. It's actually interview based. So these are two different disease concept studies that were created for Angelman syndrome. And both of these, what they do is you would have someone interview you and they wouldn't ask you any specific questions at all. They would just ask you exactly, tell me about your day. What did you do this week? Tell me about your evenings. Tell me about school. Tell me about vacations. What is it like for your family? What about meal times? And so you just tell a story of your life, how the person in your family got diagnosed, all the different facets of your life. That then is turned into a true picture of the disorder. Instead of just what physicians care about, instead of just the questions that we ask on natural history, it's everything describing your life. And you all have already gotten on the waiting list to be able to be chosen by one of the students who's going to be working on one of those. So cross fingers, you have a student choose Mowat Wilson this coming year as their thesis project, and cross fingers, you'll be able to get called and interviewed and tell your story. What do we do with all this? Once we have a disease concept model, we can actually look across disorders. So this here tells us, these were the first 20 that we did. This tells us which disorders have the most in common. So we can see here that GLUT1 and PEX10 have a lot in common. SATB2 and SETBP1 have a lot in common. And so then you could start grouping the disorders about maybe putting them together in clinical trials or using the same outcome measures or having the same clinics, sharing a clinic, a center of excellence. So this is really my advice to all of the patient advocacy groups, and when they ask what should they do to try to help get treatments into patients as fast as possible. Number one, encourage hope. Art Baudet, the the geneticist who discovered the gene for UBE3A, he was always hopeful. He always encouraged hope. And really, he was scoffed at early on. People really didn't think this kind of a treatment could actually make a difference, and it is making a difference. Always encourage hope. Diagnose people as fast as possible. If you don't have the phenotypes that can be easily diagnosed by clinicians, move quickly to whole genome sequencing. Think out of the box for treatment strategies. There might be something brand new coming down the pike, lipid nanoparticles, CRISPR, some kind of small molecule. Think out of the box. Put patients in the center of all research. Fund the design of meaningful outcome measures, what matters to your community. Collect natural history obsessively, because that is where you can make the biggest difference. Fund biomarker studies. Share your data with as many groups as you possibly can. Share it with doctors, share it with researchers, share it with industry. And share your biospecimens, your EEGs, all of your studies radically, as much as you can, so that people can do the work they need to do. So this is combined brain. What we say is we're putting the pieces together, And these are all my current staff people. It's actually not current. We have a whole bunch more of them that I should be including here. I've got to add their pictures to this. And that's the whole story. Thank you so much for letting me tell you about it.
Terry Jo, thank you so much. The work you all are doing is amazing. To the community, if you have questions, please put them in the Q&A. And while we're collecting those questions, I just want to let our community know what we are doing at this point with Combined Brain. You can see there's just a multitude of things that this allows us to do that we could never have done or do on our own. So our research committee has been meeting with our medical advisory board several times now to try to identify what are the things that we need to do first. So to let you know, we have signed a contract for the biorepository, and you'll be hearing a lot more about that because we plan to do a collection at our conference next year. And that will mean that we'll also want those of you that can't go to the conference to participate in the road shows. So look for a lot more information concerning that. We also, I was just today emailing with Terry Jo that we have are signing the contract with the Rent-A-Brain, which will allow work to be done that our medical advisory board just doesn't have the bandwidth to do. So Haley Romero, who is one of the individuals that was on the slide that works with with Terry Jo, is going to be working with Dr. Vivian, and she will be putting together a landscape review. What do we know and what are the gaps around Mowat Wilson Center? There may be studies out there in other countries that we don't even know about. So we need all of that information put in one place. So the landscape review is part one of that Rent-A-Brain contract. And the other is that Haley will help us develop a strategic plan. How do we best move forward? Because as you can see from Terry Jo's presentation, there's a lot there. And we're at the beginning stages, but we're going to be able to go a lot faster than we've ever been able to go. So just some really exciting things there. And we'll be keeping you aware of what's going on. We're going to be using the research corner in our medical news, in our family newsletter to communicate these things with you. So, Jeanette, if there are some questions in the chat that we have for Terry Jo.
There is just one, and it's
Okay.
research related, but I will go ahead and read it. Has there been any research around an increased risk for, I think it's infection slash sepsis? If not, does anyone have any knowledge on the subject that they can share?
I guess that's a question for you, right, Al? In terms of, uh, I mean, have there been particular papers published on Mowat Wilson that include any kind of an assessment of risk for infection?
Yeah. Not that I'm aware of, but that's a question we can take to the medical advisory board for sure.
And that's the kind of thing you can collect in your natural history. So when you are on the matrix, you can actually do that individualized measure I was showing you. And if infection is an issue for your child, you can actually enter every time the child has an infection. Or there's a particular survey called the Clingan surveys, which asks head to toe, do you have a problem with your head? Do you have a problem with your chest? Do you have a problem? They will ask in that survey about infections, and you can say whether it seems like there are a lot of respiratory infections or skin infections or whatever they are. And once you have enough people filling out that survey, then you can look and see maybe there is an increased risk of infection.
Thank you. All right. Again, Terry Jo, thank you so much. And what Terry Jo's talked about is a great lead into what I want to talk about, because I think you can now better understand why the registry is really at the focus for everything moving forward. researchers need to have data, and that data is going to come from our registry. So, Jeanette, if you want to show we'll start with the slides just to
Absolutely.
briefly.
There was a quick question that came in that I think
Oh,
Terry
okay.
Joe could probably answer that would be important for our community. Thank you. Interesting and exciting. How is the data protected? Specifically, it's important to share, but it needs to be used appropriately to help. So I am curious.
So, the data is actually kept on a platform that is HIPAA and GDPR compliant, so it is protected. When data is shared, it is not shared with any names, addresses, or identifying information. It's shared only by a study number. There's a number called the CRID, the Clinical Research ID, and it's shared that way. So I think that's what you're getting at. So it's important for some people, they also can opt out of sharing their data. So the nice thing about the matrix is um, patients can actually share their own data with anybody they want to. You could share it with your babysitter if you need your babysitter to have your vaccination schedule or your medication list or whatever. Or you could say, do not want my data shared at all. So you can make those decisions ahead of time. I don't want to pressure you, but what I can tell you is if you want treatments developed for your disorder, sharing your data is the most important thing you can do.
Okay, that's it for questions. So Al, I'll go ahead and pull up your slides.
All right. Okay, so now that we, I think, better understand the importance of the registry, I want to just take you quickly through how you get started. So if you go to our foundation website, you'll find this page. It's the page that lets you visit the Matrix portal. As a little bit of background, the foundation started to work with Matrix back in 2020. And at that point, Matrix didn't have a registry component, but what we were working with was the ability to track all those things Terry Joe was sharing with you there. You can track medications, symptoms, activities. You can set medication refills and reminders, and you could store documents. And I think there is actually back from 2020 on our YouTube a tutorial that I did that will show you how to use all of those different uh, components to that part of Matrix. Then the other part, the registry, we added that to our Matrix in 2022. So that's a little bit of background there. So when you get to this page on our website, you hit the Visit Matrix portal, and it'll take you to this next page that you'll see. And this is where you can log in if you are already registered, or if you're a first-time visitor there, you just request access. Okay, next slide. Okay, so when you request access, you'll get the email that's down here at the bottom. Your account has been created, and please follow the link in the validation process. So you'll click on that, you'll be able to create a password, and then when you go into this back on the site, you'll be able to now navigate and do all the things that you need to do. So this is just showing a quick snapshot of when you request access, what information you will need to provide. All right. So this is just kind of the first thing that you'll do once you are in the system. You will be providing demographic information on yourself as the caregiver, whoever the caregiver is. So you'll go through a couple of pages of demographics. Jay, you can go to the next page, just again showing you what you'll be filling out. After you've done that, The big thing that you'll be asked to do, go ahead to the next one, is you'll be asked to fill out consent. And that's where a lot of our registered caregivers right now have stopped. So just to give you an idea, we have 200 registered caregivers, which is really outstanding for our community. But out of that 200, only 100 of them have completed the consent portion. And you have to complete the consent before you can participate in any of the surveys and any of the natural history in uploading genetic test results. So for those of you that have never been part of the registry, please do. And those of you that registered but haven't done the consent, please go back and do that. So once you've done all the consent, you'll now be in your dashboard and you'll be able to add a patient. And you'll get this information and then it will take you right to the consent. Then you'll be asked to do a couple of different things. So we were talking about you filled out the caregiver general information. Now you're going to do a patient informed consent. And when you do that, we also have put in there another consent for the caregiver so that if we ever do any surveys or studies for caregivers, you have already completed the consent for that piece. Then you'll enter patient general information. That's where we'll be able to start to get the demographics for the patient. And this goes back to the question we had about sharing your data. You will have the opportunity with this question to say, you may share my patient tool data. Please don't share any of my data, or please ask me before sharing data. So that's in your control. And again, remember, as Terry Jo said, the information that is shared doesn't contain identifiers. It's de-identified data. Okay. Once you've added a patient, now when you log in, you'll come to your dashboard and you'll have all of this information on the left side that's for you. And then you can click on your patient and then it'll take you to their dashboard. So we can go down. All right. So this would be the patient's dashboard. And here on the left, you see all those things we were talking about. You can put health information. You can track symptoms and activities. You can do a journal. Then the documents, that's extremely important because that's where we're asking you to upload genetic test results. So when you've gone on, registered, filled out consent, we really don't have any other surveys in there right now. The first thing we were trying to do is get people registered, get consent, and get genetic test reports uploaded. So you'll see up in that, if you go to the next slide, you'll see that you can click upload, that's up in the right corner, and then you can put the genetic test results. This would be where you could put EEG results, any of the different records that you want to have in your patient's file. And then Dr. Schreiber is hoping that shortly we'll be able to ask and start collecting EEGs. So that's another important part of what Matrix offers us. All right. So again, my call of action to everyone is to please participate. It's going to be the most important part for us to make all of these great things that we have access to in Combined Brain come to life for us. So please share your data. And I always say, because we don't know what's possible. So thank you all so much. I think everyone has my direct contact information, and I'm happy to walk anybody through. There's a brief tutorial as well about going through all of these steps individually. So if you can put up with about 20 minutes of me going over screen after screen, please go take a look at that. But also don't hesitate to reach out to me either through the foundation or through my direct foundation email, and I'll be happy to help with any of that. Any questions for me at this point? Again, we can look at Q&A or chat, anything that might come through.
Al, there's someone who asked if you can share your direct contact information. Um, we have everyone who's registered, and we're happy to share detail after the webinar today, if that would be helpful, Al, or what are your thoughts
Yes,
on
that would be great if we could just send that out to everyone.
that? Okay, terrific. We will do that. And then there's another question, similar question to the earlier question, but how does the Mowat-Wilson Syndrome Foundation protects its data?
So through using Matrix, I mean, that was one of the main things that we looked at when we selected Matrix was all of the protections that they have. And I can tell you just from what we found and the fact that, Combined Brain is using Matrix, Jason Colquitt, who runs across healthcare. I mean, he is a rare disease individual himself, so he understands all of this. So the protected data is, I feel very comfortable with. Okay.
So, how soon will they start having surveys?
So we're hoping to do that within the next month, Terry Jo. We're just trying to, with the medical advisory board, kind of get a consensus on what we should do first and how we should start to put the pieces out there. But the first thing is get the consent done and please upload genetic test results. And the surveys will be coming, as well as more information about the biorepository and collection that we'll be doing at our meeting next year. All right. Thank you, everybody, for listening tonight and participating. Again, Terry Jo, thank you. Jeanette, thanks for setting all of this up. And I look forward to some just great